Antibody-coated pathogens are recognized by accessory effector cells through Fc receptors that bind to the multiple constant regions provided by the bound antibodies. Binding activates the accessory cell and triggers destruction of the pathogen. Fc receptors comprise a family of proteins, each of which recognizes immunoglobulins of particular isotypes. Fc receptors on macrophages and neutrophils recognize the constant regions of immunoglobulin G or immunoglobulin A antibodies bound to a pathogen and trigger the engulfment and destruction of immunoglobulin G or immunoglobulin A coated bacteria. Binding to the Fc receptor also induces the production of microbicidal agents in the intracellular vesicles of the phagocyte. Eosinophils are important in the elimination of parasites too large to be engulfed. They bear Fc receptors specific for the constant region of immunoglobulin G, as well as high-affinity receptors for immunoglobulin E. Aggregation of these receptors triggers the release of toxic substances onto the surface of the parasite. NK cells, tissue mast cells, and blood basophils also release their granule contents when their Fc receptors are engaged. The high-affinity receptor for immunoglobulin E is expressed constitutively by mast cells and basophils and is induced in activated eosinophils. It differs from other Fc receptors in that it can bind free monomeric antibody, thus enabling an immediate response to pathogens at their site of first entry into the tissues. When immunoglobulin E bound to the surface of a mast cell is aggregated by binding to antigen, it triggers the release of histamine and many other mediators that increase the blood flow to sites of infection. It thereby recruits antibodies and effector cells to these sites. Mast cells are found principally below epithelial surfaces of the skin and the digestive and respiratory tracts, and their activation by innocuous substances is responsible for many of the symptoms of acute allergic reactions. Have a great day.